Good morning, everybody. I am most excited and pleased and happy that we are starting our first PhD student conference in ecology at the Faculty of Life Sciences. And we say the first, with the idea that this will become a tradition, hopefully, in the next years. So I am most pleased for you all coming to this conference. And we actually, when started the idea, Abed, our dean, came and to me and asked if I could be able to organize, and I was lucky to recruit or we have been working together since then. The idea of getting exposed what we are doing here in our faculty. I think we are doing an outstanding research in ecology, environmental sciences, and I think we have a chance of knowing each other. Many students don't know each other from different schools, and this will be, I think, starting of a tradition of getting know each other. So with no further ado, because of the little delay, I'm happy. With no further ado, I hope I'm put here. I will speak louder in the next. So with no further ado, I'm happy to ask our dean, who's Abed Salem, to introduce us to work.
Thank you, Marcelo. So I'm very glad to open this first students' ecology meeting with the Faculty of Life Sciences. We hope it will not be the last one. I hope we will have more meetings for that. As in all events, usually the organizers ask me to say a few words. This is good because I don't need to talk too much. So I will introduce one main feature of our faculty. So if you don't know, Tel Aviv University constitutes of 9 faculties, and we are one of them. And actually, if you look in terms of economics, we are recruiting 25% of the budget of the university. People do not know that. They think that other faculties in the east side of the campus do that, but we are responsible really for the quarter of the income of the university. This is we think also that we are providing the best teaching and producing the best research, and this is due to a decision we made long time ago that we are keeping all disciplines of biology strong. So we are the only faculty in Israel that keeps zoology, botany, and molecular biology equally strong. There is no other faculty in Israel that is doing that. And of course, ecology is central aspect for our faculty. You will see that in the talks today, and you will see what we are really doing excellent research in ecology and of course other subjects related. So I would like to thank Marcelo and Bob for coming with the idea. And of course, I would like to thank the students that will present, I'm sure, excellent talks and excellent science. Good luck.
Thank you very much, David. First of all, I also would like to make some acknowledgements at the very beginning, because this effort has been also joined with our faculty and Moran and Laurence, are been helping us and being behind scenes and trying to organize this event the best way possible. And I hope you will see it soon. The title, when we look on the title, the ecology in the area of global change, we're actually in these presentations we'll be able to see a variety of topics that includes what we call it global change. We live in an area of the time of global change, and this includes land use change, sea change, and definitely global climate change. So with no further ado as well, we start with our first presentation, the first student presentation. Sorry, I just want to show you slightly the schedule with few delay, or almost no delay. We have our first talk, Shlomo Praise Bloom, who is going to introduce us his work. It's a student from the Marta Young Lab. Then we have Merav Level from the What's Appeared lab, Vicky Barzor, Vicky Barzipso from Or Spiegel lab, Lachan Roth from Omri, and last for the first station, Tal from Shai lab. So we have a variety of topics that you can see here, a variety of subjects, and then we, after a coffee break, we continue to our second session, understanding ecological services with novel tools, and we have another 5 presentations. Then we have a short break, and then our last and third presentation. Sorry, we have launched our keynote speaker presentation. Dr. Noam Leader from the Nature Conservation Authority is going to share with us his experience of doing ecology on the field, on truth and coming from the academia, after finished with his PhD, what he found when he was doing real work in an organization as NPI. Then we have our last session with another very interesting presentations from different labs, from Amir, from Gal's lab, Ruth from Michal Goodmans, Galbert from Noah Shankar, and Ofri from Yossi Yovel. So we have again a wide variety of presentational topics, and looking forward for the excellent presentation. So we will start our first session. Sorry for that. We have here Shlomo. The floor is yours. Thank you.
All right. Good morning, everyone. My name is Shlomo. I'm a PhD student of Tamar Dayan, and I'm doing my PhD on the ecological aspects of human-wildlife conflict in the Golan Heights, looking at how human activity and trophic interactions are shaping the mammalian community. Today, I'm going to present the first chapter of my PhD, which we recently submitted for publication. So we'll start with a quick introduction about wolves in the Golan Heights, and I chose to frame this globally. Today we're talking about global ecological changes, and globally, move this Zoom bar out of the way so you can read the titles. Okay, so over the past 50 years or so, large carnivores are recolonizing human-modified landscapes worldwide. We see this happening in Europe, where bears, wolverines, lynxes, and of course wolves are naturally recolonizing areas where they were hunted to extinction over hundreds of years of persecution. In India, tigers are increasing in numbers in human-modified landscapes. And sometimes this recolonization is also actively done by humans, like the famous case of wolves being reintroduced to Yellowstone, as you can see on the bottom right. And when large carnivores return to human-modified landscapes, it inevitably brings with it cases of human-wildlife conflict. The most common source of these conflicts are damages to agriculture, whether it's elephants raiding palm plantations in Indonesia or wolves attacking livestock in the United States or in Israel. Okay, so the Golan Heights is no exception to this rule. The Golan was recolonized by wolves after being annexed by Israel in the early 80s. Wolves, ecologically speaking, are our area's last remaining apex predator. This has significant ecological implications for the entire community. And since being recolonized in the 80s, today the Golan has a small but very dense population of gray wolves living in between 9 and 11 established packs. That's the last estimation of the Nature and Parks Authority. They've been doing so well that they've expanded into the Galilee and the Carmel in the recent years, Galilee in the early 2000s, Carmel really recently. And what makes the Golan so interesting is two things. One, we're a few steps ahead of areas in Europe that wolves are recolonizing today. So we really are sort of a case study for these other places that are undergoing wolf recolonization today. And two, if you look at this map of land cover and land use, just notice how many different colors there are. Red is military firing zones. Green is nature reserves. Orange is land mines. Purple are croplands. Here we have a very, very dense matrix of land uses, which is something that is unique to this area. And as you'll see later on, the data that we have on the wolf population is also very high resolution, which makes it really unique and interesting to study. Our population of wolves here in the Golan subsists both on wild prey, such as young wild boars, gazelles, hares, as well as carrion and livestock and garbage from human sources. I can verify this because now I'm doing a scat survey, so I've seen it with my own two eyes. And there is a lot of local knowledge about this wolf population. The Israel Nature and Parks Authority monitors the population closely. Ranchers that we work with know these wolves very intimately. There hasn't been a lot of scientific publication in the last 20 years, so that's where we come in. And this population of wolves in the last 25 years or so is found in conflict with livestock ranchers, because wolves will do what wolves will do with free-ranging cattle, and that is eat them. So in the context of this conflict, the ranchers want to protect their livelihood. And the same way each ranch raises their cattle in a different way, each ranch handles their wolf problem in a different way. Some use lethal methods like shooting. Shooting is a legal method with a permit today, in contrast with illegal lethal methods like poisoning, which has been illegal for many years. And other ranches are using non-lethal methods, which we'll talk about. And the Israel Nature and Parks Authority, which is called the INPA from now on, is responsible with managing this conflict and managing these conservation considerations against other conservation considerations like the vulture population or the gazelle population. So they have a tricky job in managing this conflict. And some of the management solutions that exist today are, one, fencing, which has each management solution has pros and cons. So fencing, for example, it can decrease calf and sheep predation. These are animals which are more vulnerable than adult cows. The downsides is that it causes habitat fragmentation. Gazelles can't go through these fences as well as other animals. And the maintenance costs are very high. And as soon as there's one hole in the fence dug by a porcupine, widened by wild boars, The animals come and go, and that harms their effectiveness. Livestock guardian dogs are employed on some farms. When used correctly, they can deter predators. Another benefit is that they live with the herd. You don't have to send them out. They're there 24/7. Some of the downsides include attacks on wild ungulates. I have footage from my own cameras of dogs chasing after gazelles, and it really is hard to manage them. Here in Israel, the current livestock husbandry in the Golan, we don't have thousands of years of history of working with guardian dogs like they do in other parts of the world, so this is still something that hasn't quite been mastered. Disturbing wolves to make them leave conflict zones with light and noise. On the plus side, it's preventative, okay, it's not lethal. And in other places in the world, it can teach a pack to stay away from an area. However, when we're talking about an ecosystem and not only wolves, these can provide noise and light disturbances to a system that could otherwise be natural. And lastly, the method that we're going to talk about mostly today is culling by shooting, which on the plus side gives the ranchers the feeling that they're being listened to. It's an immediate response. And ostensibly, when the ranchers feel like someone's listening to them, they're less likely to go and put out poison and devastate the vulture population of Golan. Some downsides of shooting wolves is that it can disrupt their social structure. Wolves are pack animals, and other studies have shown that when wolves are shot, then they're actually more likely to attack easier prey because they can't hunt wild prey anymore. And culling has also been shown to be a short-term solution. Unless you wipe out the entire population, the population will recover and visit the neighbor's farms. So, as I said before, here in the Golan, we have really a luxury of data, very high-resolution data of exactly where and when wolves are shot, exactly where and when depredation occurs. So if you look here in the red, we have cattle killed by predators each year. And in blue, we have the number of wolves culled. So you can see here that the INPA implements different solutions, and each time there is a dip in the number of livestock killed. We've seen that the wolves in the Golan are very adaptable, and if you try to find a correlation between the number of wolves killed and the number of livestock killed, there is none. It's sort of it's a management plan, and what we want to do is understand what this is doing to this wolf population as apex predators in the mammalian community. I put up here a picture of many dead vultures because this actually all started here in the late 1990s from the big mass poisoning event of vultures, which of course was aimed towards wolves and jackals, but killed mostly vultures. So one thing I want you to think about is really these competing conservation interests that the INPA and we as researchers have to look at. So the first research question is really, what is this culling doing to wolves in space and in time? Here is a map of where and when wolves were culled. Darker spots are more recent culling, lighter spots are longer ago. And as you can see, the culling is not uniformly spread in space. There are areas that have higher culling in them. So assuming that the risk of mortality for wolves is different in different areas, I wanted to see how wolf activity was different in these different areas. We added to that the question of land use to see how different land uses affected wolf activity, as well as the interactions between the culling and the land use to see if culling affected wolves differently in different types of land use. So the method I use is camera traps. That's where all these nice pictures are from. I set up a grid of 60 cameras. You can see here on the right, each dot is a camera across the Golan in 4 types of land use: high culling in red, low culling in green, orange minefields, and nature reserves blue. This is what a camera looks like set up. And for each camera spot, I recorded environmental data in this app called Ecology. We collected over 1 million images. We used Microsoft's AI to get rid of all the false triggers and then identify the species manually. We recorded 20 different mammal species, of which 6,000 images were of wolves. So for the spatial analysis, I used a really cool function called focal, which instead of just measuring the proximity of a camera to a certain type of land use, it also takes into account how much of that land use is within a given radius. So not only how close I am to a nature reserve, but how big that nature reserve is. And that gave me environmental data for all of these different spatial layers for each camera point. Now, I use this function also to create rasters for the culling data itself. Okay, so for each camera point, I really got the culling intensity of how many culling cases there were around that point and how far they were away. I used a package in R called unmarked, which is used for modeling unmarked animals that we don't have individual identification. Most of the pictures of wolves were at night, so this was the right package to use. And I ran two types of models. One, occupancy models, if a site is occupied by wolves, yes or no. And two, count or abundance or activity models, which take into account the relative abundance or the density of the activity at that point. And we found some really interesting results, which we recently submitted and should be hearing back any day. So the first thing we found was that 57 out of 60 cameras recorded wolf activity, okay, almost all of the cameras. And as you can see here, the background, the red background is the culling intensity. The circles, the size of the circle is determined by how many wolves were recorded there. So really wolves are everywhere in the Golan, more or less, and they're a very mobile species, so that makes sense. However, their activity levels were impacted both by culling and by land use. We found that culling had a positive correlation, and I say correlation carefully, with wolf activity. We found that nature reserves had a positive effect, open space, cattle presence, these are things that we expected to see. Infrastructure, meaning towns and roads, had a negative effect on wolf activity. Minefields surprised us, and we suspect that it was the case. As you can see, if you look at the Syrian border on the east side of the map, only the top minefield had a lot of wolf activity. The southern two had less wolf activity, and the INPA thinks that it's a case of unreported culling. And then we found an interesting interaction between culling and open space, which I'll get to here. So first, I thought it would be really interesting to separate between single wolves and pack wolves. Okay, this is data we had from the cameras, and you can see here that the blue circles and the yellow circles denoting pack and single wolf activity are not in the same places at all. The single wolves were most active where culling was strongest, and the packs were most active away from these culling zones, which was really interesting to see. The top graph shows that single wolves had an almost linear relationship with culling, meaning it didn't matter what type of land use it was, they were attracted to these culling zones. However, the packs, as you can see, in the most open spaces, the blue line in the bottom graph, culling had a negative effect on pack activity, and in red, in less open spaces, culling had a positive effect. Temporally, here we have clocks. Okay, this is a clock showing how the times of activity of single wolves, wolf packs, and human events. Human events could be a hiker, a car, a bike. And first of all, we find a predicted result, which is that humans are active during the day, wolves are active at night. That we know from the literature. Secondly, we see that there's no significant difference between single wolves and wolf packs. What we do see in the right column is where culling is the most strong. We see that wolves are active exclusively at night, whereas in lower levels of culling, you can see here, they utilize morning hours between 5 and 7, early evening hours, where culling was less intense, but where culling was most intense, they were really limited to late night activity. So just to summarize what we saw, one, wolves were really ubiquitous across the landscape. They're all across the Golan, but at varying intensities, meaning our activities are affecting how wolves are using the landscape in both space and time. Importantly, culling did not deter wolves, especially single wolves, from these conflict zones. Again, I don't say that the culling is causing high wolf activity. We're noticing a significant correlation between culling and these wolves. Packs were able to utilize these protected areas. The single wolves were not. We see this sometimes in other places in the world where packs control good territories, and the single wolves have to find their own territory, so they take risks. And in conflict zones where the cattle is easy to find. All the wolves, regardless of whether they were packs or singles, shifted to nocturnal activity under heavy culling. And this, of course, has implications for management policy, both here, where we can kind of almost predict We know that the single wolves are being more attracted to these conflict zones, so we can adapt our policy to this. And also abroad, we are a case study for other places where wolf recolonization is just starting now, and we can really provide lessons for these places where this is happening. In the next chapter of my research, I'm going to be broadening my focus to see how the wolf activity, which is being affected by human activity, is affecting other species such as jackals, wild boars, and gazelles, and affecting the wider ecosystem. So thank you to my supervisors and co-authors, and if there are any questions, they have to
Thank you very much. We have time for only one quick question.
So for the average day, how far will these packs be traveled? What kind of distance will they cover?
That's a great question. So my project student Neely actually did a project going over our wolf images, and she identified packs and individual wolves. So we actually see that there are lines of territories that they tend to not cross. Average roaming distance is 7 kilometers in a day. Average pack territory size is about 80 square kilometers, which is small compared to the world. But those are the distances that we see. It also it depends on the season. When the cubs are small and in the den, they tend to venture less. And when the cubs are older, which is now what we're seeing, they'll tend to travel a little bit further.
Thank you very much. Good night. So, just a short remember to the speakers. We have in total 15 minutes. Actually, we would like to have some minutes for questions. So try to remember 13 minutes. And when you reach 13 minutes, I would like you to sign. So please, Merav is going to present her PhD. So no further ado.
Hi, and so my name is Merav, and today I'm going to present to you my PhD work on quantifying the potential need of endangered plant species for improving overall introduction success. So,
קליק עם עכבר ואז זה.
Okay, so as probably most of you know, global biodiversity is threatened by human-induced processes, mainly changes in land use that was already mentioned, and that causes habitat loss and fragmentation. Because of that, we have to use some conservation efforts, including in situ, inside conservation, ex situ, and in some cases, when in situ conservation is not an option or is not enough, we do range reduction to protected sites. Range reduction is the intentional movement of an organism, usually inside its native range or its habitat. And it's not necessarily to the sites where these species lived before and disappeared. In many cases, they are not available anymore, those sites, natural sites. But we have to remember that we have a limited resources for conservation, that being money, time, human resources, and also seeds, because many of the endangered plants are very rare, they are very small populations, and we don't want to damage them by collecting a huge number of seeds. So we have to do some prioritization and decision making. We do this with the Natural Parks Authority. And a red list of endangered plant species was published. We have more than 400 plants that are endangered in Israel. This is a prioritization list. And from this list, more than 50 species were already reintroduced in the last decades, but mostly without any scientific research. In some cases, we have monitoring for a few years. In some cases, we don't have any data. And that means we can't really learn why did we fail or succeeded. Here are examples of 3 species that were reintroduced with a proper research. The only 3 ones published in Israel. You can see they are all big, shiny flowers. 2 of them are irises. So what about all the other species? In my work, we're trying to improve the reintroduction of endangered species using distribution models as a conservation tool. I will show it later on. And also performing the reintroduction experiment in the field, and then trying to compare the reintroduction success to the model's predictions. So what are species distribution models? SDMs take data collected in the field, mainly presence and absence data, and then environmental layers, and uses a statistical model. There are many different models you can choose, and try to predict the potential distribution of the species, and we get a potential distribution map. It was already used in some cases to try and find the actual niche of the species, mainly with species that are in big areas and we don't really know the distribution. It was also used to try and compare between species. It was used to try and predict response to changes, for example, climate changes, and also to predict invasions of invasive species. It was also used in conservation, mainly to try and locate areas that are fitted for new natural reserves. And in this work, we suggest to try and use them to try and predict the success of reintroductions. If you think of the potential distribution map, the niche map or the species potential niche. So if we get a model with the best available niche, we can think that maybe as we choose the better niche, the reintroductions success will be higher. Of course, it cannot be the case if, for example, there are other factors that are affecting most the reintroduction success. So I also want to say it's very important when we talk about endangered species, they are usually very rare. Models don't work very good with low amount of data. So here is an example of a very rare species, Woomexer palaniformis, Kumata aviron. It's endemic to Israel. It grows today only in 2 natural sites, and in all the history we have records of 8 sites. That's not really enough. But what we do have is good data about past reintroduction sites from the last decades, and we try to use sites that succeeded reintroduction after 10 years as presence data, and we use it to teach the model. And here you have an example of a potential distribution map. In red, 1 is the highest possibility, the best niche available by the potential map, and the dark blue are sites that are less fitted by the model production and prediction. Here is another example. We can also use SDMs and try to project for future climate change using models that already exist of climate change. And here is a Salvia egi, another endemic species to Israel. On the left side, you can see the distribution map, the potential distribution map as it's today. And on the right side, you can see the future projection with climate change in a scenario of business as usual, okay? So in all the species I checked, the projection to 2040 gives a map with the lower potential distribution sites. You see the reds are disappearing. And in this case, you can also kind of see a trend of movement into the north. It doesn't happen in all the species, but in all there is a decrease in the potential available niche of the species. So for the introduction experiment in the field, I chose 2 endangered habitats in Israel, sandy soils and loamy soils. Israel has a very dense population. You can see this map with a big area, the agriculture, and most of it is concentrated in the coastal area. So we're left with very small nature reserves, mainly in the Sharon Plain, that are sandy soils reserves. And for loamy soils, the case is that there are no natural reserves at all. Because of a long history of human presence, those lands are mainly in the valleys that are used for agriculture. So when we look at these nice maps, we have to remember that all the red sites, many of them are not really available for introduction or for conservation. So you have to take into account the what's left. Okay. So how endangered are those species? These are 3 examples from the 2 habitats I presented before. Elkanah Galilea and Elkanah Galil has only 3 isolated sites with less than 100 individuals total in all the sites together. And none of the sites have 50 mature individuals, which is like a key number that the IUCN says for a population to be viable, we need 50 mature individuals. Just to compare to birds that are reintroduced, so vultures are estimated with 2,000 individuals in Israel, and as you probably most of you know, there is a big reintroduction effort for the vultures. Another example, Verbascum beliteum, Mocin beluti, has only 2 very isolated sites, one in the northern Israel near Liman, and one in the Sharon, and together they have less than 50 mature individuals. So we really do need to do something for those species to not disappear. I also want to say that mammals or birds, animals, when you reintroduce them, if you're not putting them in the exactly right site, they can move. In plants, in most of the cases, that's not true. They can't really move, so you need to pick the right niche, the exact right niche. So how do I do it? We grow the plants here in the botanical garden. We also propagate for seeds for the animals. And I grow the perennial plants in Akfar Yarok with Ron Shushan and in Tzipori. In 2022, we started reintroducing the perennial plants, and it continued last year. And last year, we also started reintroducing the annual plants using seeds that were propagated here in the botanical garden. And here are the 5 species that were chosen after we did the germination experiment, and we chose the plants that cooperated and we could grow them. So we focused on endemic plants of Israel. That's important because then the data we have is their own distribution, and it's easier to do a whole model, a distribution model. And also we have very good and accurate data for those species from recent surveys. But we had some problems. So Elkanat Galil that I talked about before, we have very low amount of seeds. We have only 1 introduced site, and we have a refuge garden with a backup population in Tzipori that collapsed last year because of the disease. So all the plants that we prepared needed to go and be planted in a refuge garden, and I changed it to another species of loamy soils, Onosma gigantea, Sultan anaki. So as you see, they are very rare plants, and in the case of Elkanat Galil, I didn't give up, but it's not going to be part of my page. So how do we do the reintroduction? Each year, for the Salvia, for example, I chose 4 reintroduction sites, and the next year 4 more. In each site, I chose 2 different plots, trying to also find the micro niches inside the site. And about 40 to 80 plants in each site were planted. In the case of animals, so here you see works of Planiformis, 1 Shushan with beds of seeds you could propagate. So we had a total of 20,000 seeds, and they were seeded in 6 different sites. It's a loamy soil plant, so along the coast in different ranges from the when I choose the experiment sites, as I said, not every site is available, and we have in this case of the soil of the sandy soils, it's easier because we have some reserves. But in the case of the loamy soils, I needed to find cooperation with landowners, with farmers, and that's very important because I needed to find communities or landowners that would give me a land for the experiment, that they wanted to have an experiment in their lands. And also I work with the expert gardeners, Roni Shushan from Akfar Yarok that I said before, and also Uri Ender from Tzipori, to try and have the best plants to maximize the success of the reintroduction. And also some active management has to be done because loamy soils are traditionally agricultural land. So in some cases there were intensive cattle grazing, so I needed to fence, and I did also some manual grazing to prevent from high competition. So I will show some preliminary results. I would say I'm monitoring those reintroductions every year, and I still have 1 more year of monitoring. But here we have an example of Mocin beluti that was already reintroduced before by Akfar Yarok. And you can see here the model prediction. And you can see, for example, in Liman and Bnei Zion, where the natural populations also exist. they had very high prediction by the model, but not very high survival rate. I must say that for plants, survival rate of about 30% is pretty high. But you can see Kadima had pretty good results and a lower prediction by the model. I can think of different reasons for that, but you have to remember it's only 4 sites and only 2 years of monitoring. But for example, for this species, we suspect that shade is very important. So that's why last year I planted it in shade and sun in different sites. I will show another example of Salte Egi, the sites from 2 years ago. So all the sites from 2 years ago had very, very low prediction by the model, and they did pretty good. And I think that can also teach us something, because you can see all the red area in the north, it's not available at all. So we use the sites that were available. Last year, I could use sites that are at higher prediction by the SEMs. But I think maybe in some cases, the plants are where were left. And sometimes we teach the model and it uses a site that doesn't exist anymore, and maybe it's not even suitable anymore. So the idea is to use those models to improve them using the data collected in the field and to get a better picture of the available fish. So what next? We want to explore and adjust more models using also the data collected in the field. We still have 1 more year of monitoring of all range reductions, and I'm also adding soil samples to try and find a different aspect of the niche. And the idea is to generalize an approach that will help improve range reductions, because this is supposed to be an applicative work. We're trying to make conservation better, to help the decision-making people to improve their decisions. If they have only 2 available sites, what's the best from the 2 available sites, not the nice maps with the red dots that don't exist anymore. So, and of course, another thing we can do with the models is try to predict future extinctions, because we are living in a changing world, and that can help us see where what sites will be better in the future. So I want to thank all the communities, the kids, the elders, everyone that came to water the plants that were dying because it was too hot. And my 2 supervisors, Dr. Logal Sapir and Professor Rami Beneker, the Sapir's lab, the Beneker's lab, the undergrad students that worked with me, Maya and Eden, Ronnie and Uri, that without them there would be no plants, and of course Margareta Volchak from the NPA that made all this possible, and all the families and communities that took part of it, and my own family too. So thank you for listening.
Thank you, Mara, for a nice presentation. Again, we have time for 1 question. You can go. We have time for 1 question.
צריכים לך מקומות לשתילה? תמיד, הדוקטורט שלי נגמר תכף. Okay, thank you.
Can you directly incorporate availability into the model and then have model predictions more accurate from the first place?
Yes, we can do that, and it's very complicated because we have to think of what to use. Current availability, future availability, but yes, we can play with that. I tried to do land use in the model, and it didn't really help me to improve the models. It gave, because I think the land, I think the layer are not good enough, but yes, but I think after you do the model, you can put the layer of land use and then try to find what's available.
Thank you.
תעשה היי זהב.
So I'm pleased to introduce Nikki. Is from our lab. It's going to introduce. We're moving from plants to birds, and the elements that relate to behavioral selection. Please.
Thank you. So, hello everyone, I'm Mickey, and I study how urbanization affects the movement and behavior of sparrow lapwings, which is these beautiful and elegant birds that you can see here on the screen. So, lapwings are ground nesting birds, and the population in a whole of ground nesting birds in the world is declining because of the urbanization. But this species, it seems that its population is increasing. And the reason that the population of ground nesting birds is declining is because urbanization taking more and more of the land. And a lot of species getting hurt because of it. So, this is it's not right. It starts.
אפילו ב...
So the reason urban environment is such a threat is because it's such a stressful environment for a lot of animals. And this is because it has a lot of different type of pollutions, a variation of different threats, and not just cats, also vehicles can be threats. And of course, there is a lot of human interactions. So acclimating to urban environments requiring to have a unique set of behaviors that animals in the wild are not necessarily having. For example, animals in urban environments were presented to have bolder behavior to an approaching threat. And this is something that we actually already tested in the lapwings. And we wanted to see if the reason lapwings are bolder in urban environments are because of habituation to human or something more. So we tested the lapwing between two models, one human approaching and one jackal approaching, as you can see here in this video, between three main habitats. And we found that both to human and both to a jackal, lapwing allow them to approach much more. So, so this means that probably lapwing are not habituated to human, and there is something more to this. One of those options that can be is that their behavioral type is different. And a behavioral type is when an individual repeats the same behavior for a long period of time, but it's different from the population variation. And this is the testing with repeatability. With higher value of repeatability, the more you can differentiate between individuals. And there's a lot of different behavior that can be tested, such as how aggressive the animal or how shy it is. But what's interesting is that those repeated behavior are also being shown in the movement of the individual. And this is because the movement of the individual is affected both by the internal state of the animal and the environment itself. And I will give this plot as an example. So the x-axis is the months, the y-axis is movement index, and this is from the study of Herter from 2020. Each line represents a different element. And when we look at the yellow line, you can see that through the year, the line change and the behavior change. But if you compare it to the other elephants, it's still lower than the others. So we can assume that movement index can be represented as behavioral types, which bring me to my research questions that were first, can we determine behavioral types through the lapwing movement pattern? And second, if we do they differ between urban and natural environments? So to do that, I captured lapwing for 3 years. I captured 211 lapwings in Harod Valley. which is a northeast valley in Israel, which have a mosaic kind of habitat. You can find there fields or fish ponds or urban environments, which is a great place to study the system. And after measuring the condition of the lapwing and bringing it, we added an atlas tag on it. So one word about how atlas is different from a GPS, both take the location, but the atlas, the tag itself does not transmit the location. The location is calculated through antennas that our lab established around the valley. And in each antenna, there is a unique computer that tests the location of the tag. And once the bird is between 3 or more antennas, his location is calculated. And this allows us to have the location of the bird in a high frequency for a long period of time. And just for an example of how it's look, this is the movement pattern of one lapwing for 155 days. And I wanted to differentiate how the daily movements are different. So let's take for an example one day to look at. And you can see that first, there's location that the lapwing is stopping to. There's also location that is staying for a long period of time. And also you will see now the location that he started is going to revisit. And you see there's the flying pattern, there's the maximum flying pattern. And overall, when you take, I take from those movement patterns, 9 unique daily movements that I could measure for repeatability. So I will not go through all of the movements that I measured, but here on the y-axis, it's all the movement index that I measure for repeatability. The x-axis is the repeatability value, and everything that is beyond the red line is presented as a behavioral type. And you can see that all of those behaviors can represent for behavioral types, but I wanted to see how the combination of those behaviors change between urban and natural environments. So I added them to a principal component analysis, and this is the V plot for the PCA. And this will reduce all those 9 index into 2 unique behaviors. The first behavior, which is the x-axis, is the mobility of the bird, and the higher value gives me more flying and more location that's on the ground and higher distances. And the y-axis gives me the exploitation of the bird, which higher value represents more visitation to the same location and total time spent on the ground. Testing for those 2 behaviors for repeatability also were significantly repeatable, and now I can use those 2 instead of all of the 9 as behavioral types. So summarizing this part, I took the combination of all the 9 index and saw that they give me the mobility and the exploitation of the bird. And now I can see how do they look between urban and natural environment. But now the question is how do I define what is the habitat of the bird? Because as you see, and as you saw in the video, the lapwing do not stay in the same place. It's mobile, it's a bird. So to actually see how the habitats differ, I first calculated the home range of each lapwing with kernel density. And this is what you see here in this picture. In general, kernel density gives me the estimation of the size the bird is using. And the yellow color is the 95% area that he's using. And the red color, otherwise called the core area, is the 50% area. So on the home range, I added the habitat that I wanted to test. And from the core area, I extracted the habitats used. As for example, this bird in the core area used 77% of the area in urban environment. I did that to all of the lapwings that I tested, and I plotted them on a 3-way axis. Each axis represents a different habitat between 0 to 100, and each dot is a unique lapwing. So, and you can see the arrow represents the lapwing that you see here in this picture. And now that I have those results, I can differentiate between what is the habitat of the lapwing and actually test it and how it's affected by the behavioral type. Combine the 2 results that I got. And when we do that, let's see first the first behavioral type. So when we look at the first behavioral type, the x-axis is the urban and natural environment. And you see that the lapwing in urban environments tend to fly more, to be more mobile. One of the reasons for this is because urban environment is more stressful. So they do not have the time to be too much in the same place. They need to move from one location to another. But when we look at the second behavioral type, which is the exploitation, we also see that urban environment lapwings are more exploitive than the natural one. So, but interesting, I also saw that there is an interaction here with the weight. And when you look at the x-axis is the weight, the different color is the urban and natural, and the weight only affected the urban lapwings. With the more the lapwing weighted, the more exploiters they were in the environment. And I remind you, exploiters mean he revisited the same location again and again and stayed more in the same location. So summarizing this part, urban lapwings tend to be more mobile and more exploiters in general, but we also see that bigger lapwings are more exploiters in urban environments. So what does it actually mean? So first, urban habitat is more stressful. As I said, it means that the lapwing do tend to not stay in the same location because there are more vehicles and humans that go around and bother them. But if they are in urban environments, they prefer to exploit the environment better, and they do manage to find more food. And those that find better resource of food probably are better exploiters in urban environments. So in conclusion to this study, our world is becoming more and more urban. And this is something we cannot change, even if we want. And it is important to understand how animals will adapt or acclimate to those changes. And the first step for understanding that is knowing how the animal behaviors is different for animals that are in the urban environment and what other things that affect it. Like for hero, for example, it's not that the behavior, it's also the weight that affect the success of urban environment. Doing that will help us get better plans to actually preserve better animal when the urban area will naturally expand to the natural ones. And I would like to thank everyone that is on this list. I will not go through all of them, but I had a lot of help in this field. And of course, thank you everyone for listening, and I will be happy to answer your questions.
Thank you very much, Mickey, for the nice presentation. We have time for the questions. Alito, please.
What are the differences in terms of food sources? Are there different sources of food and availability of them between the urban and the?
So lapwings tend to eat bugs in general, so there is not really a much different that should be in a natural environment have more bugs in general. But we do see that the lapwings that are managed to acclimate better to urban environments are bigger. So they probably do find better resource of food, probably garbage or something that attract more bugs, and they attracted to those locations. I don't know, I didn't check what is the different type of food sources, but because they eat bugs, it's not supposed to be a problem too much for them to find the resources of food.
Very interesting talk, thank you. I have one question. In urban versus wild environments, how do you, or do you differentiate between like urban, like lawns and vegetation that would seemingly be wild, and opposed to like streets and pavements and things like that?
So yeah, urbanization, it's a level. It's not really, you cannot really say this is urban, this is not, especially in Israel. So I took like more of a gradient. I said, okay, field and pond, this is something somewhere that they encounter less human, this is less urban, and kibbutz or the other areas are urban. So this is how I differentiate between those environments. Of course, there are other ways, and there's also the human footprint that you can use, but lapwing use such a small home range that human footprint will not really give you any results. And so I differentiated by those habitat. Then other different things that you can do. Of course, you can actually check with cameras how many humans go around this place. You can check the vehicle. And by the way, vehicles are not really a problem for laughing. They do not seem to bother by them. It's more of humans that bothering them.
Just, I have one small question. They are territorial, aren't they?
Yes, they are.
How the territory issues came into the area.
So they are territorial, but they are also very social. And they are territorial in the aspect of in the nesting season. And usually in the nesting season, they do separate different nests, and then you can and those that actually in urban environments are you see less ducklings there. So maybe this is one of the reasons this is a better place for them, because there are less disturbance from other birds. Although if there's disturbance from humans, maybe humans is a less of a threat for them than other birds or other animals, or such as jackal, for example. So they're but in pond you can see them everywhere, and you see a nest every 50 meters.
Thank you very much, Vicky.
No problem.
Now we're moving from birds to the sea. We include also marine ecology.
Okay.
So without further ado, we move to Lahan's presentation of using this red alert and hurricanes in the Red Sea. Please.
Okay, hi everyone, good morning, thanks for coming. My name is Lahan, I'm a PhD student in Oceanic Monitoring Lab. Today I will be talking to you about this big title of regional scale sea urchin mass mortality in the Red Sea. So this big title is mostly composed of two main components, sea urchins and mass mortality, and I'll try to break it down a little bit. So first we're talking about sea urchins. These are echinoderm animals, and together with 4 other classes of sea stars, sea cucumbers, sea lilies, and brittle stars, they compose the entire phylum of echinoderms. And echinoderms in generally and sea urchins in particular are considered a phylum characterized by population dynamics of booms and busts. So fast increases and decreases in their population over relatively short periods of time. And their favorite food to eat is algae, so that makes them grazers on different reefs, and it highlights their ecological role on coral reefs in particular. They're basically the mongoers of coral reef, where they really graze and control the level of algae on the reef. And when talking about this triangle of love between sea urchin, algae, and corals, when everything is quite balanced, then everybody lives happily together. Algae and corals. What happens when we remove something from this equation? For example, removing sea urchins from the ecosystem, nobody else controls the level of algae, so they continue to grow and they create an unbalanced situation in the ecosystem. And since they're really good competitors for different resources, the environment, in the sea, such as for light and sediment placed on the reef, they really compete, they really beat in the competition corals, and eventually don't allow them to really grow nice and easy. The other part of this title is mass mortality, which is defined as a catastrophic collapse of a population with extensive regional consequences. And when putting these two components together, so sea urchin mass mortality, we often come to see a ecological phase shift occurring on coral reefs. You can see here in this photo a beautiful coral reef from the Caribbean. And what happens is when sea urchins are gone, often, as I said, nobody controls the level of algae. And these reefs are altered from coral dominated to being algae dominated and eventually continue to degrade if nothing else happens in the ecosystem. So I've spoken a bit about sea urchins, but today I will mostly be talking about a genus of specific sea urchins, Diadema. You can see here in these photos. These are highly conspicuous animals, so it's very easy to detect them. That really helps us to see them on the reef and to monitor them. And they actually consume a really large amount of algae. So actually no other species of sea urchins compare their ability to facilitate the algae level on the reef. And now I'd like to take you to a tour around the world, exploring some mass mortalities of different species from this genus. So the first event I'm going to talk about started in the early 80s, where Diadema antilabrum, a specific species, experienced a mass mortality event of up to 93% mortalities in the entire population in the Caribbean Sea. That's a lot. It caused a macroalgae increase in the area of up to 50%, followed by a massive coral decline of up to 80% in coral cover. Following some years later, another species to the west coast of Africa experiences another mass mortality event with 65% mortality. And as time continues, these mortality events continue to accumulate around the world. Talking about specific two events from the past year, from 2022, I'll start with speaking about another event occurring in the Caribbean, which was identified to be caused by a specific waterborne pathogen, and really caused another mass mortality of the entire population. And the second event I will refer to now is the one in the Eastern Mediterranean with the fourth species on the list of the Diadema setosa. This is what it looked like, and last year it happened. What's important to know about the species is that it has invaded from the Red Sea into the Mediterranean through the Suez Canal in 2006. And this paper talking about these mass mortalities actually warns towards the end of it against the possible expansion of these mortalities from the Mediterranean and into the Red Sea. So it was proven that these two populations in the Mediterranean and in the Red Sea are genetically the same. So we're talking about the same species, and therefore there are there's a high risk of an expansion of these mortalities to different environments, but the same population, the same species. Sorry. So this is what we know so far about mass mortalities of this genus. Today, I will be talking about an ongoing and a current event of mass mortalities in the Red Sea, which means that the warnings from the previous paper I was talking about really came true. So the way we explore these mortalities is through surveys. We go to the sea, we put on our diving gears, and we explore what's going on through performing bell transit, which is basically putting a measuring tape and measuring in a certain area the abundance of this species. So in that, we have a routine monitoring program, which every 6 months we go and we monitor different sites in Eilat, in the Red Sea. We also do that from time to time in Aqaba and Jordan, and our last time monitoring over there was in 2010. It will be important in a second, I'll explain. And I would like to take you through a walk of time and explore the chain of events that we've been witnessing in the past few months. So the first reports for these mortalities started in December 2022 in Aqaba and Jordan. And these were the evidence that we received and saw when we went there as well. So massive piles of scattered spines of these urchins, and these are their skeletons after their degradation and death, basically. In January, we received some reports from Eilat, where sea urchins were disappearing from sites that they were highly abundant before. And we obviously immediately went there to explore. And the visuals are pretty much the same as what they saw in Aqaba. And we went and did surveys again. So we were able to actually have a very unique data set of only a month apart between before the mortalities occurred and right after the mortalities occurred. We also went to Aqaba again and explored the reef over there. So the results are presented here. We can see that on the left columns we're describing the data for Diadema setosa, and on the right column we observed another species mortality of Ecology calamaris, which is a related species to Diadema. They belong to the same family. On the y-axis, for the left, we have the abundance for Diadema, and on the right for Echinopteryx. And the blue boxes represent the data before the mortalities, and the red ones represent the data for after the mortalities. The numbers here represent the decline of the population at the different sites in Eilat and in Aqaba, and we really see that the numbers match other mass mortalities events around the world, reaching up to 100% of decline in some sites. So pretty concerning. I think you'd agree. Reports continued to arrive in February. We got some reports for South location in Egypt, and then for the first time in April, we observed evidence for these mortalities outside of the Gulf of Aqaba and in the main body of the Red Sea. As you can see there in the map. Sorry for that. We went back in April to Dahab in Sinai in Egypt, and we did some more belt transits to explore the reefs again. And we see here on this measurement that a massive pile of dead urchins were documented. And when we came to quantify this data, we saw a significant increase in the number of dead Diadema cytosum. We can see that here. And we performed these surveys over a period of 2 days. Okay, so we're talking about a 48-hour difference between a very low number of dead individuals to a very high number of dead individuals. So think about how fast this thing that's happening to them is occurring. Moving on in time, some more reports came from Ras Shaitan. We went there to observe, and finally we had 2 important reports from other locations, southern in the Red Sea and outside of the Red Sea, near Oman and the United Arab Emirates. And this really confirmed to us the extensive and the regional scale of these mortality events. So when asking what is the source of these mortalities that may be, we came up with several known factors of mortalities that are known from the literature, and we asked whether these can be happening over here with our events. So in terms of pollution, no reports were documented for pollutions or a pollution event. And actually, understanding the regional scale of these events made us think that it's probably not something caused by the pollution, which will cause a more local effect. So we kind of ruled that out. In terms of environmental effects, such as high temperatures or freshwater income, we also figured out that these probably are not the cause, because first, these events occurred during the winter. And second of all, no anomalies in these 2 parameters were documented in all the stations that are recording these environmental data. Then we thought maybe it's just part of their natural fluctuation. As we talked in the beginning, sea urchins are known to fluctuate in their populations, and they can go up and down in their abundances. But first of all, this was a very, very quick decline in population, again, on a regional scale. And in addition, this population of the Diadema cytosum has been monitored for the past 40 years, let's say, and it has documented to experience a gradual decline over 40 years. What we see here is very much not gradual, it's a massive decline or massive mortality. And we came to understand that these 3 factors were probably not what's causing these mortalities. Next in line, we tried to ask whether this is a waterborne pathogen. And as I explained in the beginning, the event in the Caribbean in 2022 was identified to be caused by a waterborne pathogen. So we decided to compare these 2 events in the Red Sea and the Caribbean and try to see how similar they are and maybe conclude from that something. So when we look at the pathologies of the 2 events, we see that very similar external symptoms occur where tissue degrades in very specific areas on the body, and they progress similarly. So waterborne pathogen was identified, as I said, in the Caribbean, and I'll show you now a video that made us realize why we also thought this is a waterborne pathogen in our case. So what we see here is a tank with one individual Echinopteryx. This was taken during the night with an infrared camera in the IOI in the marine station in Eilat. So These systems are fed with seawater from the sea directly. Nothing is filtered, everything is the same. And as we can see here, for now, the animal behaves normally until now. It was moving in space. And since a certain point, it just stops from moving in space and it starts to kind of degrade. It reduces its spine movement. And if we fast forward a little bit to stage 4 during the day, you would see, I don't know if it's quite clear, but its tissue is starting to degrade and its skeleton is starting to be exposed over here when eventually it dies. And I don't know if you noticed, but in this tank, there are 7 other individuals of another species of sea urchin. It was noted at the beginning. And this made us also realize and understand that in addition to it being probably caused by a waterborne agent, that was the only thing common to this tank and to the sea, it was very specific to a certain species or to 2 specific species. It was documented as well in the Caribbean to attack only the edema species. So we see these other species of sea urchins that are not being affected at all. They continue to behave normally. And I hope that by now I've convinced you that these 2 cases are pretty much the same. And we asked ourselves the question, could this waterborne agent thing might have transferred from the Caribbean to the Red Sea? Is it possible? We don't know yet. But if so, what will be if further expansions occur from the Red Sea into the Indo-Pacific? This really puts at high risk this entire region of many marine coral reefs to a mass mortality of the same species of the Edema cetosum, which inhabits the entire Indo-Pacific as well. So as concerning as these events might be, I think that now is the time for action. On the local level, we suggest and we start to take some actions for creating broodstock populations. We suggest to take individuals from the Israeli Mediterranean that has not yet experienced mass mortalities and to place them in a disconnected tank from any body of water to try and reintroduce or repopulate this species to its native habitat in the future if will be needed. On a regional level, we suggest to raise the awareness and we hope for more data collection from other regions in the Red Sea and beyond it. And on the global level, we are currently collaborating with several research groups around the world to try and identify if this water pathogen and what is it. And we also are trying to understand with several more organizations what are the causes and changes in the world globally that causes these mass mortalities in higher frequencies that we're witnessing these days. With that, I will conclude. Thank you for coming and listening. Thank you for my lab mates and funding resources, and for all the people who took part of this great resource that was recently submitted to a really nice journal. Thank you.
Okay, thank you very much, Lahan, for this very interesting presentation. It was in the news. You might have heard the newspapers and the media your work. So we have time for one question.
Hi, thank you. It was very interesting. Do you think because of it started with the Caribbean and then the Red Sea, it might come from the Suez Canal, from the Mediterranean?
That the expansion of these, it might have, you know, it's really hard to like put a clear answer in this one, but it might have been transferred from the Mediterranean through the Red Sea. It's likely.
And there are no evidence in the Mediterranean of mortality, or there are not?
Yeah, there are. So I said in the beginning that the same species mass mortalities occurred in the same species exactly last year. More on the coasts of Greece and Turkey, not in the Israeli Mediterranean.
Thank you very much. I'm sorry we don't have time shortly in the break for President Lahan.
Thank you.
And the ballast water, we got in the ballast water algae coming from Colombia to the Mediterranean. So it's an issue. And so our last presentation for the first section I'm happy to introduce Tal, who's going to combine moving from peptides and mutations, very interesting topic. Please.
Okay, so hi everyone, my name is Tal. I'm doing a PhD at the Sherman lab with the co-supervisor Uri Wolf. And unfortunately, I'm not going to, no? I'm not going to talk to you about the scalded snakes, but I will talk about species ranges. So these are what do you need to create a species range? So you have to have observations of the species. We have, for example, Homo gamma and his observations. And in order to create an expert drone map, you need an expert, which in this case was Shai. So what they do is usually look at the observations and the suitable habitat for the species and create a range. These ranges have high implications. Firstly, for the size of the range can determine the endangered status of the species. And also these ranges put together are used to create species richness, global species richness maps and hotspots, and are used in many studies. And these two things have high implications on conservation. But we do have knowledge gaps regarding the species ranges. So if we have, for example, the range of the snake, this is what we know that its range is, but actually this is its range. And this is called the Wallacean shortfall, which we have a gap in knowledge on where the species exist. So there is a,
I thought
so there is a special sampling bias globally. You can see here a high density of ecological research, and that most of them occurred in North America and Europe. Not surprising. Okay, and we have huge spaces in the world that are unstudied at all. And for example, most of these desert areas. So if you go back to the comment diagram, you can see that Shai decided to leave these 2 points separately, and because he doesn't have enough data, wasn't confident enough that his range goes all the way there. So actually, the comment diagram can have a species a range much bigger than what we see. This variance in sampling was connected to a few predictors, few variables. So economy and safety, where rich countries and safer countries are have more sampling. Of course, roads and proximity to universities have an effect. And also, as researchers like to study endemic species and within protected areas. What was less looked at are endemic factors like freedom, education, and government, which of course can have an impact on how much research and recording of animals is done in a country. I'll just say that one of the reasons they didn't look at it is because the special resolution is usually very high. So it's more of a country-based variables. So what I want to do is to map globally the rectangular shortfall and to explain the variance in this in the knowledge with anthropogenic factors. So what did I do? So for each species, I'll show you example of one species, and I did it for all. I took its range and the countries within its range. We have, for example, manipulated. And I calculate the expected knowledge by area. So if manipulated is half of its area in Syria, I would expect half of the observations to be from there, so on. And then I looked at the observed knowledge by record. So you can see more 92% of the records are from Israel. While the other countries have very few observations. And I gave each country a score based on the observed divided by the expected knowledge. So here, for example, for this species, Israel got 46, where Iraq got 0.3. And for Israel, for example, from this species, we got this score, and I did it for
all the species we have in the data, and took the median for each country. And this I call the contribution to racial distributional knowledge, this score. I have approximately 3,500 species, because I couldn't choose species that are currently in one country, only 2 or more. And we have around 2 million records. So I took records from GBIF, the Global Biodiversity Information Facility, which takes observation from museums and research and so on, citizen science, and also from GAR, the Global Assessment of Racial Distribution, which is a group formed by my supervisors, and they collect many records and also expert drawn maps. So how do they do that? They take maps and observations from field guides, from research, and from crazy people that go and search for reptiles in Afghanistan and Kashmir, places like that. So they have a lot of data, much more extensive than GB. Okay, so because I gave the scores based on neighbor analysis, and because I compared neighbors, I also wanted to account for that in the model. So we have I use geographically weighted regression, which gives a global model, but also for each country, it creates a model for its and its neighbors. I looked at many predictors, and I divided them into groups. So for example, accessibility, research, government freedom, economy, and education, and so on. And I chose only 1 or 2 of each group because of high collinearities. So for the results, this is the global reptile central format. So gray countries are got the score as expected, blue higher and red lower. So we can see some thing we would expect, like Canada and US to have very blue color, and Western Europe, and Central African countries, and some of them are very red. But you can also see there's high variation, which is the stuff that no one has shown before, within the areas. Okay, so as I said, I looked at many predictors and did model selection, and these were the ones that by far were most important and predict this variance. So we have civil liberties, which is freedom of movement, freedom of press, everything. Government effectiveness, which how government implement policy. I asked ChatGPT to explain it for a 10-year-old. He said their government effective governments are make their people happy, because people have good lives with a lot of opportunities to grow. And the third very important predictor is healthy life expectancy, which estimates how long will a person live in health. Okay, so we're going to move one at a time. So of course, as the government effectiveness was higher, also the contribution to distribution of knowledge and the score I gave each country. There was a little bit of variance in the slope around the world, but it was pretty much important everywhere. And I chose an example. So you can see here in the Eastern Europe area that it has a very high correlation. So of course, in Western Europe, people have opportunities to do whatever they want and then go and look for reptiles. And in Eastern Europe, where the government is very corrupted and we just have less opportunities. But for example, we can also see Georgia, which is a pretty good government, and also the score again. Healthy life expectancy, also of course when it was higher, the knowledge was higher, and the white area has no significance. And if you look, for example, in our area, in the contribution to distribution of knowledge, you might have think that Iran will get a low score, but they actually got a good score, and also they have a very high healthy life expectancy. So wouldn't always what we think. Now it's civil liberties, also a little bit, it was a little bit lower in power. And if you look, for example, at this area, we can see that China has, for example, in comparison to Nepal, a very low civil liberties. And so I think that for this predictor, it's not only that there is no, it's not just that there is no data. There can be a lot of data in these places, but they're just very close to themselves and they don't really share it. Okay, that's short now. So to conclude, money can't buy you biodiversity knowledge or happiness if you don't know. But from a lot of research before, they always look at money, at GDP. And in my results, it was really not the most important. Of course, there's correlation, but I think because I looked at neighbors, you find things that you don't find if you look at in a low resolution. So in Africa, for example, in the central, of course, the GDP is very similar and low, but to the others predictors they have a different score, and it's not as important as others. You might ask yourself about accessibility, because I think most people that do field work, they see that they do it near the roads, but maybe in higher spatial resolutions, it has more effect. But when you look at the country level, it's definitely not. And education, I call this presentation what we turn after education. From early analysis, I thought that would be the stronger predictor, but since then I finished and it's not. We saw special variance in the reasons for why there is a sampling bias. So it means more room for change. So the government doesn't have to completely change in order to have more knowledge. Of course, it can be a bad change, but let's hope for the best. And there is much more to investigate. I only started working on it. I have many ideas, and if you have an idea for me, I would be happy to hear it. That's very interesting.
Take the time for selecting presentation. We have time for good questions.
Do you think there is any connection between the original biodiversity and the results? Because I guess one of Uri's and Shai's earlier work showed that there is relatively high biodiversity in the regions, or the more desert regions. Do you think this can bias the results or make these stretch more intensified?
I don't see. I did just as good as in the model, of course. I don't see a relationship, because the species mixes is higher in Brazil, and we got a low score, and in the desert we also have a low score, so I don't see it. Because also I think that the way I did it for each species had its own score giving, and
Yeah, but then a country that has more snakes has more opportunities to get scores, basically, no?
I think actually when you have more species, the median will be lower. The mean might be higher, but because you need to cover so many species, it can be a problem. So maybe it will go the other way around, actually.
Another question? Thank you very much, Tal. We are going to our first coffee break. We have a coffee break in the area between the buildings here in the backyard. So we have time, and we'll meet here in 25 minutes. It's fine with everyone.
And but then there's 3 gives and dogs.
Yeah, the presentation will give us the talk.
האמת שזה מעניין.
אני מציעה
ככל שיש תפקיד יותר גדול, זה סופר יותר דברים. יש אוכל, אל שמדביק לא היה. במי הלבין?
מה קורה?
כאילו לא רוצה להם דיפיקציות של פעם. אני מציע לעשות יותר ביולוגים תקופת העמות.
מסקנות אמיתיות זה לא תירוצים למה לא עובד. היה לי נעים כבר להיכנס באמצע, אמרתי אני אחכה להפסקה. אמרתי מה אני אפתח דלתות וזה, אני לא יודעת כדי כך מרוויחה. גם הייתי בדלתים זה לא נורמלי. שעה וחצי.
גם אני.
מאוד עשו. חסמו את כביש כל מה שמערב. ממש חסמו אותו מולי ואני כאילו...
לשעתיים ו20 מרחובות כאילו עם הצירה בתוך המסגרת שעתיים 20 כאילו סטנדסטיל, כאילו ככה, פשוט ככה.
אפילו, אבל היית מהרגע שעה הזאת, כאילו כמה שחק.
נראה לי שאני אפסיק לומר
אני עוד שנייה... גם צריך כמו...
או, אתה מבין, אני חודל כבר איזה דרך.
וזה גם גורם לכל האזור כאן להיות כזה פשוט, זה נורא.
ואז האוטובוס עוצר באמצע הצומת, ואז מישהו רוצה לתמוך שמאלה לא יכול, וזה פשוט כאילו מכוניות עומדות בכביש חצי שעה בלי לזוז.
ראיתי שחתמו גם פה, זה באמת כבר השתתפתי, כי חתמו לי 20 היה עמוד בצורה פסיכודלית, עברתי לקק"ל, ואז חסמו לי גם איזה, אמרתי, אני אערוד פה מסתיים, אני לא יכולה.
יש פה פרי הוא 3, הוא אחרון.
מה אני באה ברגל מאוד עכשיו, אני לא...
ואנחנו בקרב עירי מהשכונה שלנו, זה ממש כמה מטרים. אני צריך לספר לסיבוב על זה בשביל להגיע.
עכשיו בכלל אני כבר ברגל.
בגלל החום.
פשוט אין לנו בגלל
לא, זה בגלל
בבוקר על הקפה כדי להגיע בזמן.
גם היא אותו דבר כמו זה. לא, בזום היית יכול להקליט במשרד שני, זה לא בעיה.
לשנות את ה...
לשאול את זה.
לשנות מלעשות אקסטנשן
לא, אין
רגע, חסר לי עוד הרצאות לעכשיו. ויש הרצאה שלי. מלא? אתה דורון? רגע, מי השאיר את הדיסקונט שלי? יאללה. דורון אתה ב3, נכון? ב2, נכון? כן, ב2.
כל הכבוד
חלוקת קשב לא של טייס אבל אוקיי כן, יאללה, אני נותן לו את השם שאתה איזה מספר? 4
אילונה יעקב במקרה? לא.
אתה רוצה לבדוק איזה סרטון או משהו? רגע, תמצא אותי.
אולי תחברו את הסאונד של המחשב. סופר.
אבל יש שרוצים, אני מניח.
כן?
אוקיי, אז אתה בסדר?
סליחה על האיחור, אבל לי תמונה. הכל בסדר. כן, אני נכנסה לפשוט.
איזה אנטישמית.
כן, זה גם לא עצמם. לא נורא.
רגע. קחת את זה, זה רון?
בוא נשתה קפה.
לא, יותר מה...
אבל אנחנו קפה, זה
התיק לא נפטרים ממנו.
כן.
תודה.
צלם, אבל כבר אנחנו חלק מהדברים.
יופי.
שניהם. אתה סרט?
של החרדות, נרגיע חרדות.
אתה בסשן 2, נכון? ננהל.
כן, הוא מספר 2.
לא רוצים, מה רק שם
רגע.
אז אמרנו, עושה משהו של...
זה הפוינטר.
לעיתים.
מה?
לא, זה עובד, הבעיה היא שברגע שאתה עושה את זה, את הפעולה הזאתי, אז אתה עכשיו לא בפאורפוינט, אתה בזום, אז אתה צריך לעשות קליק אחד על המסך, לחזור לפאורפוינט ואז זה עובד. הבעיה היא לא בזה, הבעיה היא רק אם אתה בתוך הפאורפוינט או בתוך הזום. לא, הוא משתמש, כן. שווה יכול להיות יותר user friendly. רגע, בוא ננתק לך את זה שיטה בכל מקרה. רגע, זה אני רק מתן, זה השם שלך, נכון?
כן.
אז זה 3 אמרנו?
2.
כן,
טוב, חשמל שעושים.
בוא נראה, יש סרטון?
גדול. הם אמרו
שקופית 3, זה הבעייתי, זה
צריך
ריבוע, עוד לא, כן,
מה זה אדום לבן? זה פשוט בשער של האוניברסיטה.
אוקיי, זה עובד על הקצר.
זהו?
בהצלחה.
וואו.
אירופה.
אין לי בעיה, יכול. כן.
אתה
לא מכיר?
כן, אני פה, אני קורא.
יש לו קטע.
עומר, אז את הראשונה.
וואי למסיס.
אני יודעת איך לפתור את זה, אבל אני לא אסבך אותי.
לא, בואי,
אני
יש
פשוט
הפסקה.
צריכה להרצות דרך הזום.
בואי, אין בעיה, בואי ננסה למצוא לך פתרון שתרגישי בנוח לתת הרצאה. שנייה, אני אפילו לשחרר את עומר.
כן.
סרטונים.
מסתכל רגע, אין לי סרטונים, אבל שזה אפקט שאני רוצה לבדוק.
שעה זה, אין לי סרטונים ואין לי אפקטים.
כן, לדרווין היה איור אחד בספר שלו, זה בכל זאת היה לו אימפקט.
זה אפילו גנטי מוזר עם הפיצולים האלה.
בוא'נה, זה גנטיה.
זה הייטקס. את האמת, זה שלכם. ספציפית האפקט הזה. אוקיי.
לא יודעת שפאורפוינט יודע לעשות דברים כאלה.
אם אתה מלמד אותו. מסתבר.
מה, היא הלכה לכאן? ספסתי?
אין בעיה.
לא, היא רק משתנה.
זה הדיסק שלך. אוקיי, סשן, עומר, ואז רננאל, מ3,
אני חושב שאני...
ונוס זה גם ספר 3. אתה בטח פתוח, לא, אז תבדוק רגע את מה שאתה רוצה. כן, תכף אני אתן לו לעלות. תעשה 3.
תפתח אותו. שלחה את המאמר?
לא, אני נגד את ההורים, תשלח לנו. שרמן עכשיו ראיתי כשאתה יודע לפתוח את הקריירה, תן לי גם אותה לא ראית. תשלח אותה, אני מניח היום.
מה אתה רוצה לעשות?
נקשר את הסרטון, שאם אני לוחץ עליו, פשוט תכניס אותה למצגת.
יש לך את הסרטון פה?
הוא בתיקייה ליד.
שתפוק אותה, קח את הקובץ, שים אותו בתוך ה...
גדול.
אבל זה?
אם אתה לא יודע, אולי נעשה את זה. אפשר לבדוק אותו אחר כך, אני פשוט איזה פנסיטיבי.
בוא נמחוק אותו אחרי ההרצאה.
אז אפשר לראות שהוא
אוקיי, אז יש לנו... עשית סייב? עשית שיש תעודת נחש? לא עשיתי סייב? רגע, אז מה זה? 5? מסופוטיק? מי זה מסופוטיק? לא, כן. סבבה, אז אנחנו ערוכים לסשן הבא. אוקיי, את רוצה לנסות? יאללה. רגע, את בסשן 3, נכון?
נראה לי שלישה, השלישית
רגע, אבל לא העליתי את המצגת שלך עוד. יאללה, נתחיל את זה. יאללה, לך לאכול. אני אנסה איתה? כן, כן, או לא.
אני אקרא לה את האנשים.
רגע, יש לכם דיסקוטיון, אני צריכה להביא את הדשאים.
לא, יש לי. איפה התיק שלי?
שמה, אני לא... אני יכולה גם לבוא בפסקה הבאה, רוצה?
מה שאת רוצה, אין לי בעיה. אני אחלק עם פרק.
אחרת לא יהיה את הזמן להתקן. בוא בפסקה הבאה. מים,
קולה. הדיסקונטי שלי לא פה.
not judging, man.
אני
גם צריכה למצוא.
בסדר, תציעי דיסקונטי, ואנחנו מוכנים עם ההרצאות. הנה, ליאור, רגע, היא רוצה כן לנסות את הפרזנטר מוד, מה ששאלנו קודם.
לעולם?
כל עוד לא יתקינו פה מסך שני. אני מתצל, פשוט תפוצץ את זה לקרוא את המסך.
ואי אפשר להגיד לו דופליקייט או כאלה.
לא, מה שאני הצעתי.
לא, זה לא עובד בדופליקייט.
אבל אם אני מחברת מחשב שלי.
אפשר לחבר, לשתף איתך את הזום, חברי בזום.
אפשר לתת כללים. הנה, אנחנו רואים את המספר פגישה ודקות.
כן, בואי ננסה רגע להזמין אותך לזום.
המחשב שלה, אני לא יודע אבל זה יעזור לך עם המחשב.
למה, המחשב שלי לא מסך?
לא בטוח שהוא נותן לך באמת עוד מסך. הוא פשוט משדר במחשב שלך מקום. זה לא נותן לך עוד מסך.
אני חושבת שזה אפשרי. האמת היא שאפשר לעשות את זה, אז ככה שנייה, אני יכולה לנסות? אני לא אעשה נזק. אפשר בזום להגיד לו... נראה אם זה יהיה...
רגע, עשיתם בסטופ להקלטה בינתיים?
לא.
נראה לי שכדאי לעשות את עצמי, לעצור את ההקלטה.